Hi, everyone. How are you doing today? My name is Caitlin, and thank you so much for inviting us to talk at this conference. So I am one of the team members that helps support the Clinical Genome Resources Patient Registry, GenomeConnect, which I will discuss today. In addition, we have a table right outside, and I will be around throughout the conference if you have any questions. First, I'm going to review what is genetic data, and then how you and your families can participate in data sharing, and finally, why data sharing is important. Alexis Morgan was supposed to join me today. She was unable to make it, so that's why her photo is on the slide. So let's begin with what is genetic data and how do we interpret genetic data. Each of us has more than 1 trillion cells that make up our bodies, and we have different types of cells, including our skin cells, our brain cells, our heart cells, and each of our cells, all of our genetic material or our DNA can be found. Our DNA, like most of you know, is our body's instructional manual, and each instruction is called a gene, and each gene codes for a protein. To keep our DNA organized, it's wrapped up in our chromosomes. So genetic data is evaluating and understanding genetic variants. Genetic variants are changes in our DNA. While most of our DNA sequence is the same from person to person, we all have variants. Some can affect how a protein works in a body, some can cause developmental or health differences, and some genetic variants are just part of who we are. We have talked a little bit about genetic variants, and we've talked a lot today about the different tests that might be able to identify changes. Some of you guys are very familiar with this from your own genetic testing journey. Karyotype looks at chromosomes, looking at to identify large or small pieces of our chromosomes or copy number variants. Microarray with zoom in look at smaller copy number variants. And then Amy just talked about sequencing. So we can do sequencing of panels of certain genes, or we can do exome sequencing, which is looking at all of the coding proteins of our genetic material. So while our ability to detect variants has really improved over the years, it can still be like looking for a needle in a haystack. It is hard to determine what variants are associated with health and disease and what variants are important to focus on for when caring for an individual. When a genetic testing laboratory identifies a genetic variant, the next step is to classify the variant. Again, we all have lots of variants. We need to understand the gene that the variant was identified in first. What is this gene? What protein does this gene code for? And how does that protein work in our body? Would a change in this protein impact how our bodies function? After understanding the gene, the lab asks a number of questions to help classify the variant into one of 5 categories. Benign or likely benign, meaning the variant is not associated with a health condition. Pathogenic or likely pathogenic, meaning this variant is disease-causing. And uncertain significance, which means we don't have enough information to understand the impact of the variant at this time. The following speaker, Jenny Goldstein, will review this process in a little bit more detail. When the classification is known, which is the case for many individuals here, participants provide information to help understand the condition, such as what symptoms are the individual experiencing and how often? When did the symptoms begin? What is the care they are receiving? Is that care helping? Again, participants sharing their health information can be really helpful in answering these questions. For some of these reasons, the Clinical Genome Research Project is dedicated to building an authoritative resource that defines the clinical relevance of genes and variants for use in precision medicine and research, and ultimately hopes to understand what genes and variants are associated with disease and how genomic information can inform clinical care. This is kind of all summarized in this graphic, and today I'm going to focus on patients. So ClinGen has encouraged various community members like laboratories, researchers, and clinicians to share genomic and health data. ClinGen also felt strongly that patients are an important source of this additional information. Patients and their families are experts on their own health and can provide much more details about their symptoms over time, their diagnoses, and their health. So ClinGen felt strongly that access to this health information could help increase our understanding of genes and genetic variants to try to answer some of those questions that Amy brought up that we still get a lot of unknowns. So as a result, 10 years ago, the ClinGen team launched a patient registry called GenomeConnect to empower patients to share their de-identified genetic and health information with publicly available databases. So how can you all get involved? Well, GenomeConnect is open to anyone who has had genetic testing, regardless of results or diagnosis. It is not gene or disease specific. The platform consent and documents are available in English, and we are currently updating our Spanish documents, and it will be available in Spanish soon. Participants enroll online and consent to de-identified data sharing, recontact, upload a copy of their genetic test results, and provide their history through patient surveys. The whole process takes about 10 to 15 minutes. GenomeConnect is very serious about protecting the privacy of participants, and the registry achieves standards for HIPAA compliance, a federal act that aims to protect individuals' privacy. This is the same standards used by your doctor's offices. So after consenting and sharing their genetic test report, each participant receives an initial health survey that is used to capture an overview of their health. The initial survey asks general questions about issues across 17 broad categories or body systems. For example, your heart, your lungs, your digestive health. Each question provides examples of issues in that system. More specific questions are then assigned based on any symptoms or categories that the participant had or has an issue with. We also have additional surveys that can capture more information regarding maybe a history of a heart defect, cancer, arrhythmia, cardiomyopathy. Overall, that initial health survey takes on average about 12.5 minutes to complete. Participants share their genetic results by uploading a PDF or an image of their results or providing GenomeConnect with the permission to request their records from the laboratory that performed their testing or the healthcare provider that ordered it. Genetic counselors on our team review the report to ensure that all the genomic data is collected in a standardized way and ensures its accuracy. Once genomic and health information are collected from patients in the registry, their de-identified information is shared with ClinVar. I know what ClinVar is, but I didn't know about it a number of years ago, so I'm just going to take a minute to describe what is ClinVar. ClinVar is a publicly available database that holds de-identified information about genetics, variants, and its relationship to human health. ClinVar is public and freely available. It is a major resource for healthcare providers, laboratories, and researchers who are caring for patients, performing tests, and researching genetic conditions. The database is submission-driven, meaning that it relies on users, on the labs, clinicians, and others to share data. This is really how we pull our resources and knowledge in an effort to advancing our understanding of genetics and health. This is a screenshot from ClinVar. You can see up top, it provides the gene and variant information. It has things like the variant name up top in the dark blue bar. It gives the overall classifications from everyone that has submitted. And in the middle, it has more details on the variant and variant nomenclature. This is a screenshot of an actual GenomeConnect submission to ClinVar. As you can see, the submission doesn't include any identifiable information, such as a participant's name, their date of birth, but it does include the de-identified information that they provided through the health surveys. Most of our submissions, most of GenomeConnect submissions appear at the bottom of the ClinVar page. GenomeConnect does not classify variants. We include the classification that was on the report, and we indicate the reporting laboratory in our submission. Because we submit the name of the reporting laboratory, this can allow a ClinVar user to appreciate when a laboratory may have also submitted to the database. So in this screenshot, we have our submission that is highlighted in that yellow color, and then 2 rows above, you can see the submission that was provided by the reporting laboratory. In this case, was GeneDx. And this is, if you expand the view and even more, this is where we can include additional kind of data that the family has provided. So we can have additional details of that participant, such as the ages of testing, sex assigned at birth, symptoms that the individual provided through the completion of that health survey. Labs do submit to ClinVar, but they often submit in aggregate, meaning if they've seen the variant a few times, they'll submit all of that at once. So they're missing a lot of these specific details, these individual details, we call them case level details, that can be really important into our understanding of what variants and genes cause disease. So why is it important for families to share their genetic and health data? Well, from our own submissions, about 30% of GenomeConnect submissions to ClinVar, we are sharing novel information or novel variants with ClinVar. Because the family took the time to share their genetic report with GenomeConnect, GenomeConnect was the first to submit information about that variant. As we just saw, in addition to providing information about the variant, we are providing more information about the participant's health history. And this can help other users of ClinVar kind of determine maybe a little bit more clear answers about what that variant is doing. So participant sharing data, it provides a great value. And to kind of illustrate this, I want to share a case example. And for the purpose of this discussion today, I'm going to call this participant Ella. Ella is a pediatric patient with a history of seizures beginning in infancy. She had exome sequencing ordered back in 2015, and a likely pathogenic variant in SCN8A, a gene associated with the seizure condition, was identified. Her parents were tested, and the variant was not found in either of them, suggesting the change is de novo or new and unique to Ella. Because the genetic change has not been seen in healthy individuals, was found to be de novo, and was expected to impact the protein, the clinical lab 1, classified it as likely pathogenic. Ella and her family have an answer for her seizures and chose to participate in GenomeConnect to share her data. We submitted Ella's de-identified genetic and health information to ClinVar as part of the family's participation. At the time of our submission, there were 2 other records in ClinVar. The first was from the laboratory that completed Ella's testing, so Lab 1. One difference between our submission and Lab 1's submission is that we included that as additional inheritance information, that the variant was de novo. We also included information about Ella's seizure history. The second submission was from Lab 2, another laboratory that identified the variant. At that time that Lab 2 submitted to ClinVar, they had classified this variant as uncertain. That lab did not have enough information to understand the impact of this gene and didn't have enough information to know if this would be a likely explanation for their patient's symptoms. After our submission, Lab 2 reached out to our team to ask for more information about our participant and confirm that the variant was de novo. For Lab 2 with their patient, and they were unable to perform that family testing. We were able to confirm by re-reviewing Ella's report that the variant was in fact de novo, and we were able to share that information back with Lab 2. This then prompted Lab 2 to reclassify their variant and update their patients with the information of this reclassification. So Ella's results and care were not impacted by her family taking time to share her results, but ultimately by them doing so enabled another family to get clearer genetic testing results, which I think it really helps highlight the power of patients sharing information, patients having their information shared with ClinVar for other laboratories to use. In addition to laboratories using this information, another such user is ClinGen. ClinGen has various expert panels that focus on variant pathogenicity, which is understanding and evaluating if a specific variant impacts health. So again, this is something that Jenny will talk about in a little bit more detail. So ultimately, data sharing is important because it can help increase our understanding of genetic conditions and can help doctors and genetic labs better understand genetic changes that cause disease, which can be incredibly beneficial for newborn screening if they're going to be doing sequencing. This increase can help identify interventions and treatments and can help individuals get clearer genetic test results. Our hope is that the more information collected, the better we will understand how genes affect health and ultimately how to best care for everyone. So participants sharing their health and genetic data is extremely beneficial to our knowledge base, but is there any direct benefits to families joining GenomeConnect? So one feature that GenomeConnect allows is connections with researchers or clinicians So if if a clinician or a researcher reach out to us, they're interested in a particular gene or variant, we can have the ability to contact our participants. We can ask the participants to connect directly with the researcher, or we may ask the family, hey, a researcher is interested. Did you have any of these symptoms? Did you experience any of this in your health history, and share that back with those studies. In addition, we like to give variants updates on their history. So through the registry, we can facilitate genetic result updates. Recontacting families about potential variant updates is something that we know is valuable, and there's no consensus on how best to do it. There are often barriers to laboratories and clinicians recontacting families over time, from just a lack of reimbursement at the laboratory level to limited contact information sometimes if a family moves. So the registry can offer one more additional way for families to remain informed about their results. In particular, through our data sharing, we can identify instances where a participant's report is out of date compared to the current understanding. Maybe in ClinVar, we see that the variant is classified as likely pathogenic by the laboratory that did their testing, but on the participant's report from a few years ago, it is listed as uncertain. That's something we can share back with families that opt to receive this information. So, sorry, I took out an example for time, so now we're in example 3 instead of example 2. This participant and their family registered for GenomeConnect, and they shared a copy of their child's exome report. So in 2017, a child had exome testing due to a personal history of intellectual disability, macrocephaly, and polyneuropathy. Exome testing looks at all the coding portions of our DNA, and they identified a variant in the gene PHIP, and this is likely an explanation for their symptoms. However, also on their report was a variant of uncertain significance in the gene Notch1. Pathogenic or likely pathogenic variants in Notch1 are associated with cardiomyopathy, specifically with aortic valve disease, which can impact blood flow. Based on the available clinical evidence at that time, the impact of this variant was uncertain. GenomeConnect submitted all the variants that were identified on the report to ClinVar on behalf of the family. And in doing so, we learned that the Notch1 variant had been reclassified to benign by the reporting laboratory. This family opted to receive updates from us, so we shared that there may be new information about the results, and we encouraged the family to reach out to their physician or genetic counselor for more information. This case highlights some of the power of recontacting families through the registry. Often, reclassifications from uncertain to benign aren't prioritized by labs or clinicians because they're not going to impact medical care or medical management. Returning this information can be helpful to clarify results, to understand if perhaps additional testing is warranted for the child or for other family members, and it can also reduce that uncertainty anxiety. To date, we have shared over 412 updates with our participants. Some updates may have impact on their care and management. I'm talking to you about GenomeConnect today. However, I want to acknowledge that this is a team effort and the work of many. This is the QR code if you're interested in joining GenomeConnect and sharing your health and genetic information. As I mentioned, I also will be at a table for the rest of today and tomorrow if you guys have any questions, want to learn more, and take some other additional resources with you. So now I'm going to pass it over to Jenny Goldstein.